My name is Ryan, and I would like to thank the organizers of CGSI for organizing this wonderful event, and also for inviting me to give not one, but two talks. So last week I talked about data structures for k-mers, and as some of you may know, CGSI faculties are asked to submit 2 proposals for talks. And some of us got away by just giving a single talk, congratulations to you, and I wasn't sure how to handle this, so I actually submitted 2 different talk proposals, and I wasn't sure if I really wanted to give 2 talks. So my second talk proposal was supposed to be a little bit more provocative, more far off, in the hope that maybe the organizers will not select it. They did, and this is it. So the question is de novo assembly a solved problem with long reads yet? The answer is no. Any questions? Thank you. Hi. Hello. So I'm actually a new group leader at Institut Pasteur in Paris, and I work on algorithms and data structures for analyzing biological sequences. So previously, my focus was about methods around k-mers and doing some genome assemblies in collaboration with other people. So those are some of the genomes I collaborated assembling. You can find me also on Twitter. And today we'll talk a little bit about genome assembly. So as Mike already mentioned this morning, so probably all of you at least have heard of this problem. But for the sake of spending 20 seconds on explaining it, so you have something to sequence but no reference genome. So the sequencer spits out a bunch of reads, which are subsequences of that genome. And the task is to reconstruct an hypothesis of that genome into contigs. It's a very fundamental bioinformatics problem that has been around for a very long time. It has many applications. The most obvious one is to reconstruct genomes or transcriptomes. Metagenomes are just genes from related species. It has also side uses which are no less important, such as finding novel insertions, or SVs, as Mike talked this morning, finding SNPs inside non-model organisms, analyzing cell-free DNA, as Faraz was talking about in the retreat last week, doing pangenomics, and probably a few other applications as well. And happy birthday, genome assembly, because it's a problem that has been standing for 40 years now, expressed apparently since the first time in a publication in 1979 by Stedon. It says with modern fast sequencing and suitable computer programs, it is now possible to sequence whole genomes without the need of restriction maps. I'll give a short algorithmic history of genome assembly. And I'm guessing that maybe some of you already have some parts of this history. And I'll start a little bit with a very fundamental computational problem called shortest common superstring. It's also an NP-complete problem. So this is the second NP-complete problem talked about this morning. The problem consists of having a set of strings and organizing them horizontally in order to construct what is called a superstring that corresponds to a consensus of these strings that is the shortest possible. Since there are no known polynomial time algorithms for doing this, then assembly was realized using greedy algorithms. Like we're talking about 20 years ago. And then string graphs and the Bruin graphs, which are more advanced formalisms for doing assembly, have actually been introduced 2 hours one after the other at a DIMAX workshop in 1994 by Gene Myers and Waterman. And I'll mention that there is actually a very nice paper that tells the history of genome assembly that was published not so long ago by Myers. Modern genome assembly is done using graphs. So to give a very short overview, so a graph is constructed where the nodes are either reads or k-mers from the reads, and edge correspond to overlaps between reads or overlaps between k-mers. So we are talking about the Bruin graphs and string graphs. And to continue this little journey in the history of genome assembly, I will start by saying that initially the problem was formalized as, in this graph, return a path of minimal length that traverses each node at least once. So computational people among you might recognize that it looks like a Hamiltonian path finding problem. We'll traverse each node, but since it's at least once, it's not exactly Hamiltonian path. It's generalized, but it's still NP-hard. So it's a hard problem. So later works have established formally that genome assembly, as formalized in the previous slide, is NP-hard. But then it's not over when you say this, because actually, if you look a little bit more closer, if you have an instance where all the repeats are longer than the reads, so imagine short reads, imagine long repeats in genome, then actually genome assembly is solvable in polynomial time, which is kind of a surprising result, but it's no less true. But what's more interesting, if the repeats are either shorter than your reads, and the longer repeats are spanned by the reads, by span I mean there exists for each repeat instance a read that is longer than these repeats and spans a little bit to the left and to the right of that repeat. And if you have this for all the repeat instances, then assembly can be solved in polynomial time and has a unique solution. So this is actually very nice, and it tells us that we can actually work with long reads and do some good work. But in practice, with Illumina data, so reads are too short, none of the previous formalisms apply for that reason, and also because coverage gaps and biases in sequencing made that the graph was disconnected. So finding a single path inside this graph was simply not possible. So because of that, then assemblers were returning contigs, which are an ambiguous path in the graph. And nowadays, with long reads, then it's maybe more possible to bridge theory and practice, because then the repeats are spanned, the graphs are not disconnected anymore, and in fact, there has been some work from the hinge assembler, which is an assembler produced by the same authors that I talked about in the previous slide. So in my opinion, it's one of the first attempts to bridge theory and practice in this area. And I was hoping that more people, more groups would be working in making assembly look more like Hamiltonian path filing, but actually it doesn't happen so often nowadays. I will mention an article that has been published a few years ago, actually last year, that retraces the history of how assembly was modeled along the years. So starting from shortest common superstring to Hamiltonian path and other formalisms. I think it's a nice story that tells you how to model any biological problem into computer science. And the example here was genome assembly, but I'm sure it could be applied for many other problems. And since many of you here work on human data, I have to mention something about human genome assembly in this talk. And that something is actually a talk given by Adam Philippi earlier this year, where he talked about the status of genome assembly for human and vertebrate genomes. And just to give a small outlook, okay, so first of all, the slides are online. If you look for the title, you can find them. So vertebrates have high repeat content. Some have higher repeat content than others. When a genome is deployed, then it's still hard to assemble. And for humans, there is hope, as Mike said this morning to the questions, that telomere to telomere, so single chromosome assembly, will be feasible within the next, according to Adam, 2 years, next few years. And in fact, recently there has been a major achievement that the chromosome X has been assembled telomere to telomere for the first time. And today, I want to talk about something different. I'm going to be the grumpy bird compared to the person who's before me. Adam talked about very optimistic scenario. Let's talk about the supposedly easy cases. Like if you have a simple genome, bacteria, that's haploid, can we at least do a good job with assembling them? Before I answer this question, let me just mention that genome assembly is actually a complex operation. So to make a genome assembler, it's generally the task of a PhD student for his whole PhD, or a team, an engineer for a few years. And an assembler generally consists of a lot of code containing not always independent components. And what you're looking at here is actually the dependency graph of files, of source code files of a single assembler. That's just a part of it. each node is a file, and each edge is whenever a file is has an include statement for another one. It's C code. And I had the wonderful idea to ask an intern to actually isolate a component from an assembler. That was this assembler represented by this graph. And then she came back to me and she said, well, this assembler has 400 files. The component requires at least 100 files. I don't know how to do this. It seemed easy as a research question, but in practice it was really hard because assemblers are extremely complex. And on top of that, there are heuristics everywhere in order to deal with specificities of the data. And I won't read it, but there is this quote that I like from another developer of an assembler. There are quite many genome assemblers for long reads that have been released. That's not exactly exhaustive list, but I think it's close to it. The last one, Peregrine, the paper has been released a few hours ago. So it's really a booming field, I think. And I'll talk a little bit about the status of these assemblers. So a few years ago, an hypothesis was emitted also by Adam. So 1 chromosome equal 1 contig. Is it possible to actually achieve that today? So if we look at the assembly graph of a bacterial genome, if you construct an assembly graph with a small k parameters, then you see that it's a complex graph. It's hard to assemble. But if you increase k, which is related to the read length, then you see that after a certain threshold, all the repetitions are resolved, and the graph consists of a single node corresponding to the sequence of the genome. So the argument here is that if your reads are long enough and your coverage is good enough, then assembly should be doing perfectly fine because you are jumping over all the repeats, and then there is no difficulty. So that was the hypothesis. Is it true? If we look at a database called NCTC 3000, it's a database that sequenced a lot of bacteria using PacBio. So each entry in this table corresponds to a sample, a bacterial sample. And if you look closely, there is in this column here, an assembly was made for each of the sample, and that column shows the number of contigs obtained per sample. So if everything was well, then that column should contain only ones. And it's the case for many samples, but not all of them. And so actually there are 30%, 34% of assemblies which are not automatically single contig in this database. So my observation is that based on this, apparently assembly is not fully solved. If it was, then we would have 1 contig, 1 chromosome assembly for each of the samples. So to give an idea of what a failed assembly looks like, we have simulated the bacteria genome with simulated reads and assembled it using Canoe, which you heard about earlier this morning. And the resulting assembly looks like that. There are 3 contigs. And the contig graph is not very informative because it doesn't tell the adjacency between contigs. The question is, can we recover like adjacency information, so missing edges in this graph? So we attempted to do that. But before we tried to do that, we wanted to see if it was a problem due to some repetition inside the genome we tried to assemble. So we did a dot plot of contig versus genome, genome versus genome, and noticed that there is a large repetition of actually around 440 kb tandem inside this genome. So it's kind of hard to resolve, even using long reads. And the breaks in the assembly correspond to extremities of this repetition. But there is an extra break between contig 8 and contig 4, which is not inside a repetition. So why is that? To see why is that, we actually constructed an overlap graph of the reads, of the row reads. And so the nodes of these graphs are the reads, and the edges corresponds to a high confidence overlaps between those reads. And then we annotated it manually and noticed that regions of the graph which are a little bit complex, such as this one, corresponds to repetitions, this one as well. This one was correctly handled by Canoe, and you see that the contigs, actually I forgot to mention that the colors of the nodes of this graph correspond to contigs. So here, the assembler correctly assembled the contig and resolved the repetition. Here it's interesting because there is 2 different contig extremities, the red one and the green one. Apparently there are links in between them, but first of all, the assembler did not correctly process that junction and produced 2 different contigs, but also it did not report links between the two contigs. So we were left with some information that was present in the raw data, but that was not present in the processed data. So we thought about automating this type of analysis, and we produced a pipeline. A PhD student, Pierre Marijon, produced this, and he was here actually last year at CGSI. So he took a pipeline that takes assembled contigs, raw reads, then does some classification to remove plasmids and so on, constructs a raw string graph, and then does path search between contigs using the string graph. In the end, it produces what we call an augmented assembly graph, and then we can actually perform some analysis, same as I explained before. So the augmented assembly graph looks a bit like this. It's a graph that is undirected and weighted. The nodes correspond to contigs, extremities, and the edges correspond to either extremities of the same contig or paths found between two different contigs in the read graph. So using this formalism, we can go from an assembly graph that is quite uninformative because it has no edges between nodes to a graph that looks a little bit like this. So we still have the same contigs, but now we know the adjacency or the putative adjacency between contigs. For instance, these two overlap, and these two are separated by a very long segment, which actually corresponds to the instance of the repetition I talked about earlier. So we applied this technique for actually many data sets, 38 of them, and on average, Canu returns 4.3 contigs per data set, and that corresponds to around 5 dead ends in the assembly graph, and we were able to resolve roughly half of them using information present in the raw reads. So it shows that there are some progress that can be made in assembly using this type of techniques. So we went a little bit further and looked at what augmented assembly graphs look like. So they are generally complete. Like this is an example here. You have contigs and their extremities, and then you know what is the putative adjacency between that contig and any other extremity of any other contig. So here, the green one corresponds to the solution, and you can see that the adjacency corresponds to 18,000 bases. But you could also jump 100 kb and jump at the extremity of the other contig and so on. So that produces a complete graph. And then we can finally do something that theorists like, is to apply a nice theory and try to see what are the Hamiltonian walks in this complete graph. We can enumerate them. It takes exponential time, but it's still possible. And then we can weight each of these hypotheses, and we can assume maybe that the lowest weight walk corresponds to the true genome. Because it would kind of make sense if it was the case. So here, if you look, the green walk in total, since there are overlaps which are considered to be zero weight edges, the green walk corresponds to a low amount of bases, and another random blue walk corresponds to a longer amount of bases, so it's unlikely to be the true solution. So we did this for a large amount of data sets, and this plot is a little bit hard to explain, but on the x-axis is solutions ranked by their weight. So this is the best solution we found, second best, third best, according to their weight. And the weight of the solution is actually on the y-axis, and there is a diamond somewhere on the plot, here, over there, and there, whenever the solution is the correct one. So our hypothesis for the assembly is the correct one. What we want is that the diamonds are all to the left. That would mean that the lowest weight Hamiltonian path corresponds to the true genome. What we observe, unfortunately, is that for some of the data sets, the correct genome solutions correspond to a Hamiltonian path, which is not exactly the lowest, but close. In some cases, this is true. And in fact, some of the diamonds here are overlay each other. In some cases, this hypothesis is true, but not all the time. To sum up this part, bacterial genome assembly is not fully solved in my opinion, but we can construct computational objects that help. And in fact, we had this project initially for doing metagenome assembly, but we noticed that even single genome assembly was not fully solved. So we went back to single genome. So Pierre has released a software called Knot that for a single assembly produces a very nice report, a quite comprehensive report about everything you can tell about your contigs and how they are linked to each other. I'll also mention that there exist other tools that are not based on graph, but that can also perform assembly investigation, for instance, Tapestry. On a similar topic, and in fact a topic that is very related to what we talked about at the retreat, benchmarking. So how robust are genome assemblers? It turns out that a few months ago, Rayan Chikhi has released a software called Badread that benchmarks assembly in the following way. It generates reads which contain either chimeras, low-quality regions, systematic base calling errors, in order to kind of simulate what the data what kind of badness can be in the data, and see how the assemblers react to this badness and if they are robust to it. And for those of you which have done computational studies, this looks a lot like software testing. So you might know what the unit test is. So it's when you program a function and you write another one that is a test for that function. For instance, if you write an add, you want to make sure that add at least works for doing simple things. It's different from a functional test where you actually don't want to test a function, but a whole feature. So for instance, if you want to test a read mapper, then you can call the mapping algorithm on a simple read and a simple genome and make sure that all the results are consistent with what you expect. So you're basically testing a larger portion of your program, not just a function. So people did assembly functional testing. In fact, Rayan did. So what he took, and it's published on his GitHub, is he took a single bacterial genome, pretty much like I did in my previous part of my talk. And that genome has a pesky little tandem repeat of 20 kb of rRNA operon. And then he simulated reads using Badread with various read length, error rate, coverages, and various other artifacts. And now that's really a nice benchmark in my opinion, and the results are here. So he took a few assemblers, a few software, and each of the points in this graph is a single assembly. You have to keep in mind that generating assembly is quite expensive, so it took a lot of CPU hours to do this, I guess. In this graph, the x-axis corresponds to the simulated read length, and the y-axis of each of them corresponds to contiguity. So the higher, the better, and in fact, when it's 100%, it means that the genome has been assembled as a single contig. So an immediate observation is that assemblers generally perform well after if you have reads of length 20 kb, which is not so surprising because that was the longest repeat inside this genome. But some actually are able to perform even better, even with shorter reads. And there is some interesting jittering in some assemblers where, in fact, with high coverage, they don't even achieve perfect assembly in some cases. But some are actually much more robust. So that was for read length. There is also examination of read depth. So if you want to know how deep you want to sequence a genome in order to have a nice assembly, then this provides the start of an answer. So assembler seems to perform well after, let's say, 25x. But then again, the threshold really varies depending on the assembler. So here, WTDBG and Fly are able to assemble at something like 15x, which is quite low sequencing. But have to keep in mind also that this is simulated data. So in actual data, there might be some more variance in read depth. And another plot which I find quite interesting is read identity. So we know that depending on the chemistry, then the read accuracy can be high or can be a little bit lower. And it turns out that assemblers, the ones which have been tested here, all perform well on current chemistry error rates, so around 90%. But assemblers like Ra can even work with much higher error rate. So we talk about benchmarking. And what about benchmarking the benchmarks? So I wanted to reproduce the results of Rayan and see what was my experience with it. So I took the same bacteria as Rayan, and then I simulated reads using another read simulation program, a very recent one, PASS, at 50x depth. So 50x, you should be able to assemble a bacteria with no problems. It should assemble fine into a single contig, maybe. And what I'm showing here is just the read depth at each position. That's 50x. You can notice that there is a small thing here. That's higher depth of coverage. Why is that so? That is the repetition of 20 kb inside the genome. So it's a tandem repeat, so you can see that there is a higher depth here. So apart from this, it's pretty much nicely uniformly covered. I took the same assemblers as Rayan. And can you guess what the results are going to be? Drum roll, please. It's not always perfect, surprisingly, even at a coverage which supposedly assembles well. So after that, I was asking myself, is this a coverage problem? So then I upped the coverage even more. So at the very high end, so 100x, there should be no problems here. Of course, there is no problem. There is. It's actually still problematic. Depending on the software you run, you might not have a perfect assembly. So I'm using number of contigs as a proxy, but it's really, at this type of instance, if you don't have a single contig, something is wrong. So it's not a coverage problem. So I wanted to test something else, like robustness to coverage drops. So taking the same data set as before, and then simulating a nasty little coverage drop of 10x. So I took a region inside the genome, so maybe I think this is a 10 kb region, and I removed manually some reads from this region to see how robust assemblers are to this type of artifact. Can you guess what is happening when I do that? Nothing. Assemblers are actually extremely robust to this type of coverage drops. So it's actually the same. And I think WTDBG knows even better. No, it does the same as before. So nothing happened. Let's be extra evil and even lower the coverage to 5x. Nobody in their right mind will assemble at 5x. So that region here should definitely produce some sort of contig break in many assemblers. Will it? Not all the time. So I would say good job, Fly, for being able to jump over a 5x coverage drop in a simulated setting. And so after that, I decided that it was enough benchmarking the benchmarks, and I stopped at this point. So going back to my grumpy bird conclusion, the status of genome assembly is not fully solved, even for bacterial genomes. Benchmarks are, of course, very helpful for doing this. And today, I presented two tools that contribute to it, one from my PhD student, Not, and one from Rayan Chikhi, Badread. And now this is my actual thank you. And do you have any questions?